Hello everyone, and welcome to the Slightly Scientific Podcast. I'm Oliver, and today I'm going to be joined by Finley and Stacy. Together, we'll be interviewing Dr. Stephen McCarthy, who has been researching how to make painkillers from spider venom. Now, because I record my intro and outro after the interview, I can tell you that this is a fascinating topic. He talks about how we feel pain, why peptides in spider venom stop you from feeling that pain, and why some people don't feel pain at all. I think you will really enjoy this episode. Now on to the interview. So, first question, what is pain?
It's actually quite a difficult question. On the one hand, it seems like it should be quite simple, right? Because we all know what pain is. We've all experienced it. But from a sort of, not quite molecular level, but from a sort of biological point of view, it's surprisingly difficult to nail down exactly what it is. I mean, we all know it's a sort of an unpleasant sensation that you get when you've hurt yourself, you've somehow damaged yourself, or you could damage yourself, right? Like, there's a risk of that happening. But we also sort of know instinctively as well, there are different types of pain. Like, the pain you get from, I don't know, if you sleep awkwardly and you have a bit of a sore muscle in the morning is different to the kind of pain you feel when you get a paper cut, for example. Likewise, if you get a headache, that's another kind of pain that feels different to those other two types. So trying to understand exactly what those differences are when it comes to sort of what's going on in your body can actually be surprisingly tricky.
What is different about the people that you based your research on? Why can't they feel pain?
So I should probably explain. So the research I do is sort of in the field of chemical biology. And what I study are compounds that interact with a particular protein in your nerve cells that is very important for feeling pain, or at least some kinds of pain. And the reason that I study these compounds and I study this protein is because there was a study that came out about 14 years ago now. It's quite old by this point. And the people who wrote the study found a family of people in Pakistan who were apparently, had nothing particularly different about them, apart from they were unable to feel any pain whatsoever. And this is quite unusual in some respects. It's not uncommon to find people who have decreased sensitivity to pain. Some people have it, much to a very great extent, but there are other things about them which, you wouldn't necessarily want to sort of think about as when you were designing a pain drug, for example. Like there's a family in Italy that can't feel any pain, but they also can't detect any differences in temperature, so that they can't feel whether an object is hot or cold, for example. And there's another lady who was discovered a couple of years ago now. She lives in Scotland, who can't feel any pain at all. And interestingly, she also doesn't really feel any stress or anxiety for similar reasons, which is, great for her. Apparently she's very happy all the time. But equally, you want to have some emotional responses to some things that can cause stress, because that's, I'm not going to say part of being human, but it's an important thing to feel a natural sense of anxiety. It helps protect you if you're in a stressful situation. So this family in Pakistan were really quite unusual in the sense that they couldn't experience any pain whatsoever, but their motor functions were totally fine. They could feel hot and cold objects. They had, there was nothing obviously different about them apart from that they couldn't feel pain. So if you're a researcher that wants to discover a pain medication, for example, a new painkiller, well, something you can do is you can look at this family and you can say, okay, well, what is different about them in the sort of genetic, in their genetic material, sort of at a molecular level? And does that tell us something about what aspect of their physiology is means they can't feel pain. And can we replicate that in, everyone else, essentially? So researchers in this study did some genetic analysis on this family, and they found that they all carry mutations on a particular gene, which, and as you might know from school, the genes that we have in our bodies, various functions, but some of them encode proteins. So your body can read the gene and it turns it into proteins that perform particular functions. And in this case, the gene that was different about this family in Pakistan encodes a particular protein in the nerve cells. Makes sense that a protein in your nerve cells would be involved somehow in signaling pain, right? Yeah. So after that report was published, a lot of people, myself included, started investigating compounds that can affect the behavior of this protein, which all of us carry in our bodies, with the goal being that if we can find a compound that can block this protein, maybe we'll be able to recreate this pain-free state in people like you and me, who do experience pain. And then you've created a new painkiller.
You said there was like a woman in Scotland who couldn't feel stress or anxiety. How do you measure stress and anxiety in a person?
This is again, very difficult. I mean, the simple thing to do with people is you can ask them, how stressed do you feel? But in terms of sort of rigorous measurement, it's pretty difficult. Yeah. I mean, actually, pain itself is very difficult to measure. Like if you think of people who research heart disease, for example, they can measure all sorts of things about a heart. Like, they can measure your blood pressure, they can measure how fast it's beating, they can measure lots of different parameters. So they can sort of, measurably see, well, what happens if we apply this drug or something like that, and you get a measurable outcome. But even with pain and stress, there's no sort of measurable thing you can do like that. The best thing we can do for pain is really just to ask people, how intense is your pain on a scale from 1 to 10, which is pretty subjective. I think it's probably fair to say. So that's one of the challenges of making a pain drug, is that, it's very difficult to really know how much you're having an effect when all you can do is really ask people how much pain they're feeling. And that in itself doesn't encompass all of the different aspects that I mentioned already about pain. Is it like the sort of dull pain you get in a headache? Is it the sharp pain of a cut? Some people who have their limbs amputated could feel pain in a limb that no longer exists. So, how do you try and explain that with what we know about the nervous system? It's very difficult. It's very difficult to sort of measure and quantify. And it's just one of the challenges involved in making these painkillers.
Well, that is quite interesting. I do have a question, which is, what's so special about spider venom, and why don't you use like other species of venoms?
Sure. So I mentioned that I was studying compounds that can bind to this particular protein and maybe block it and prevent it from performing its usual functions, and therefore prevent people, you and me, from being able to feel pain. The compounds I study, or one of the compounds anyway, is from the venom of a particular spider. And venom is a really interesting place to look for compounds that could be good painkillers, because if you think about what the good properties of a venom are, right? What do you think about when you think of a venom, right? It's fast acting, it's really potent. You don't need very much of it, right? Because most venomous creatures aren't very big. They can't make huge amounts of venom. So if they make a small amount, then what they make has to be very potent. And these are actually properties that we really like in good drugs as well. We like them to be, very, very potent. We don't have to take very much. We like them to be fast acting, all these sorts of things. So they're a good place to look for medicinal compounds. And what's interesting about venom in particular is that a lot of the compounds that are in venoms act on the nervous system, which makes sense. You think of a venom, like, if you want to paralyze your prey, say you're hunting an animal and you want to paralyze it so it can't run away, or even if an animal is hunting you and you want to paralyze it so that you can run away and it can't chase after you. It makes a lot of sense when you sort of think about it in those terms. So yeah, there are lots of compounds in venom that act on the nervous system. And spider venom, it just so happens there are a huge number of different species of spider, tens, maybe even hundreds of thousands. And not all of them are venomous, of course, but those that do produce venom, which is a pretty big subset, will each have dozens, maybe hundreds of compounds in their venom, which have an effect on the nervous system. So it's a huge resource. There's probably hundreds of thousands, if not millions, of different compounds out there, which we can investigate and see if they have an effect. Spiders are not the only species that we investigate for these things. There are lots of venomous species that we look at. In fact, there is already a drug that is approved. You can take it if you get in a very bad way, which is derived from the venom of a cone snail. And it's called Zichonatide. So this is not just sort of a theoretical thing. People have already done it. But we like to look at spiders because, as I said, there's a huge diversity in the venom, and also because spiders, particularly the large like tarantula type spiders, they produce quite a lot of venom. And when you're trying to find compounds in venom that could have these effects, you want to find a species that produces quite a bit of venom, because otherwise you'll be there trying to extract venom from a huge number of animals in order to get enough to do the analysis. So it's sort of convenient to look at spiders in a way that it isn't really to do sort of centipedes, for example.
And
I think the spider that you studied was the Peruvian green velvet tarantula. How did you find that particular spider would be that its venom would be a good painkiller?
Well, it wasn't actually me that discovered that. It was already known when I started my research. Essentially, it was discovered because you can perform screens of different compounds. So you can take entire venom, so straight from a spider, and you can, it's called fractionation, which you may have done a bit of in science, where you separate out the different components as best you can, and then you take each one and you perform a test against the protein that we're studying, and you can measure the response. So for most compounds, they won't do anything, right? You add your compound and you'll see no change in the response of the protein. But for the compounds that are having an effect, you will be able to detect that response. So now you know, okay, well, in this fraction, I know there's a compound that has an effect. So you zoom in on that fraction and you create more of it, and then eventually you discover the chemical formula and hopefully, and then the structure, and then you have a potential compound that could be useful as a drug lead. In my particular case, so the molecule I study is a peptide. It's a very small protein. It's called Protoxin-II. And when I started my PhD, it was known that it was very, very, very effective at blocking the protein of interest. But it wasn't really known how it did that. And understanding how it does that is a really important thing to do in medicinal chemistry. Because if you understand how it is interacting with that protein, and then what it's doing to that protein in order to prevent the protein, to block the protein, you can then start to design other things that might act in the same way, but better. If you don't have that information, you're kind of sort of guessing blind, if that makes sense. So that was where my research came in. So it was sort of structural studies, making modified versions of this peptide, and then trying to see what effect that had. And also trying to work out, some information about where on this protein it was binding, so we can design newer, better drugs that, do it even better, if that makes sense.
Yeah. And those peptides you were talking about, how do you find out the structure of them? Was that the fractionation you were talking about, or is there a different way to find out the structure of the peptides?
Yeah, it's different ways. Fractionation is where you have a mixture of compounds and you want to separate them out so you can look at them individually. But that doesn't necessarily tell you anything about the structure. There are a few different ways of finding out the structure of a compound. The one I used is called X-ray crystallography, which essentially involves growing a crystal of your compound of interest. So in my case of this peptide, and then you shoot a beam of X-rays at it, This when the X-rays go through the crystal, they diffract, so the beam kind of splits, and how the beam splits allows us to work out what was in the structure of what was in the crystal to begin with. It's a little more complicated than that, but essentially that's how it works. It's quite old, actually. The first X-ray crystal structures are decades old at this point, so it's been around for quite a while. The hard part is getting your compound to form a nice crystal, right? It's a little bit harder than like the salt crystals or sugar crystals that you probably see on a daily basis.
In fact, in chemistry, we're actually making crystals. We're making magnesium sulfate and copper sulfate literally just on Friday. It's quite
fantastic. Well, the copper sulfate is actually one of the very first compounds that was studied by X-ray crystallography. So yeah, it's nearly it's around 100 years ago now, I think. They took a big copper sulfate crystal and shot X-rays at it, and that was one of the ways they discovered X-ray crystallography. So yeah, some things never grow old.
Exactly. Now, what can you infer from the structure of these peptides?
So once you know the structure of the peptide, you can see which of the sort of which parts of the peptide are where in space, obviously, and that allows you to get some information about which parts might be important for the binding and the activity against the protein. So not all of it will be important necessarily. So you want to identify just the sort of the key points where it's interacting with the protein, and then what those points are doing that prevents the protein from performing its function. So in our case, we could see from our crystal structure that it's very tightly coiled. Protoxin-II is a good way to describe it. It's very compact and very rigid, and we think that that's important because if you have a very sort of floppy peptide that can sort of move around a lot in solution, as it would be if it's in some venom, then it isn't sort of organized to bind to the protein in your nerves. By being very rigid and very sort of compact, it means that all of the sort of the key parts which interact with the protein are in one place, so that when it gets to the surface of your nerves, where this protein is, it's sort of ready, if you like, to bind on, and it doesn't have to do any sort of jiggling about to get into position. So that's one thing that we could see about it. I mean, the other thing that we wanted to do is, once we had the structure, we could sort of guess which parts might be more important than others for the activity. So if you can say, okay, well, these parts are the bits that are responsible for blocking the protein, then you can maybe design a compound that has just those bits and doesn't have the rest of it, and see how that works and what does that do about how does that behave when you apply it to the cells. And this is something medicinal chemists do all the time. It's a sort of a key part of what's called structure-based drug design. So you take a structure that works, you try and identify the important bits, and then you use that information to try and design new and hopefully better drugs.
Well, this all sounds very important. However, how can you make sure that a painkiller is reliable? And how reliable does a painkiller need to be so that people can start using it?
It depends what you mean by reliable. What do you mean by reliable in this particular context?
For example, are there side effects are there side effects? Can people get hurt? Is it safe?
Sure, yeah.
And how often does it work as well? And how many people does it work for?
Yeah, I mean, these are all really good questions. The way that we test drugs is through clinical trials, essentially. And a clinical trial is where you take a very, very large number of people, ideally, and you test your compound against whatever it is you're looking at. So in this particular case, in the case of a painkiller, you would find people who are experiencing some form of pain, or who are happy to be given a little bit of pain in the right context, and you either give them the compound that you're interested in, or you give them a placebo compound, which is a compound, or it's something that looks essentially identical to the drug you're giving them, but doesn't actually contain any of the active compound. So it shouldn't work. And then you compare the responses before and after. And if your compound is having the effect you want, then the people that took the drug should see increased pain tolerance or decreased levels of pain. And the people that took the placebo should see, they might not see the same as people who took nothing, but they shouldn't see the same levels as the people who took the real drug. And by doing it in a large number of people, you try and measure exactly these things that you were talking about. You try and see, well, how many people did it have this effect in? If you've done your job right and designed the clinical trial correctly, then there'll be a very, very diverse group of people in it. So you're making sure that it doesn't affect some groups of people more or less than others. So in terms of before you even get to that stage, there's a huge amount of testing that will go on before you get into humans. So usually compounds like this are tested first in cells. Then if that's looking promising, you might move into mice. And then if that's looking promising, you might do some other studies on other animals, things like toxicology studies, which is really where you look at the side effects essentially. In the mice, you tend to look for just the sort of effect that you're looking for from your drug. When you go to other animals, other than mice, beyond that, you're starting to look at what else could this drug be doing? What happens to this drug when it goes into the body? Because your body doesn't know it's there to help you. It's a drug. It will freely modify it and digest it and do all sorts of other things that your body is normally naturally able to do. So you want to make sure that when it does that, none of the things it produces are going to cause you any harm. And you want to make sure that the drug, when you actually sort of give it to someone, is actually getting to the place where it needs to be in the first place. So this is something that is quite important for particularly peptide drugs like the one I study, because peptides, as they're small proteins. If you give that to someone as a pill and they swallow it and it will go into their stomach, well, it'll probably just be digested, just like every protein that you eat. And therefore it probably won't get to the place where it needs to go. The stomach will digest it and it won't be any good. So you need to think about how you're going to give people the drug as well. So that peptide I mentioned that is available as a painkilling drug, Zichonatide, you have to take that intravenously. So they inject it into your bloodstream. So that avoids the problem of getting digested by the stomach, but it has some downsides because, obviously it's a much more dramatic intervention. There are people who don't like needles, understandably, and also means you can only really take it in a hospital environment, right? Where they're trained to inject things into your bloodstream. It's much more convenient to have a drug that you could take, like you would take a paracetamol, just from a bottle at home. So there's a whole field as well of trying to make peptide drugs that you can take as a tablet rather than have to inject. So these are all the sorts of things that when you're designing a new drug, you just have to consider even before you get as far as the clinical trial. So there's lots of work to do, if that makes sense.
Yeah, wow. Seems like there's so much to do before you even get to the clinical trials. And how many stages of clinical trials do you have for painkillers?
Well, in terms of that's a really difficult question because it sort of depends on what do they show. Generally, as a sort of a blanket rule, and this is not just for painkillers, this is for most drugs, you'll get what we call 3 phases, right? So you have the phase 1 trial, which is in a very small number of people. And the goal of that trial is just to see that nothing bad happens, essentially. You give them this drug and they're okay. They don't show any serious side effects. And sometimes drugs do show serious side effects at that stage. And you want to find out in a small number of people in a very closely observed, monitored setting before you start giving it to lots of people. If you pass the phase 1 clinical trial, you move on to a phase 2 clinical trial, which is where you start to look at efficacy. So does it work in treating the disease that we hope it will do? And those tend to be larger. If you have incredibly good results at phase 2, you might be allowed to not do another trial. That's kind of unusual, but it can occasionally happen. But then most drugs will go on to what's called a phase 3 trial, which is in a very, very large number of people in sort of, if you like, realistic clinical settings. So people with real diseases, with all the sort of natural variability that you find in people, in the general population. You want to make sure that you're getting a very diverse representation as much as possible, and that's really the sort of the key test, right? Will it work in the real world as much? You want to simulate that as much as possible. So generally, there are 3 stages, but some drugs have more, some drugs have less. Pain is one of those things where it kind of depends on the type of pain, right? If you want to see if you've got a drug that will cure a headache, well, that doesn't take all that long to test, because headaches don't last that long, and generally you can expect your painkiller to respond pretty quickly. Whereas if you're looking at people with something like chronic pain, which is where they feel pain almost all the time, even when there's no real injury or any sort of obvious reason for it. You hear about this a lot with people with sort of chronic back pain or chronic neck pain. It's a very serious problem. But obviously, people who experience that kind of pain, you'll need to conduct a much longer trial to see if the painkiller is having an effect. So if you take your painkiller, does it work for half an hour and then wear off? Or does it work for 2 weeks and then wear off? And these are very, very different, obviously, in terms of what you're trying to treat and what you need to do to monitor your trial. And yeah, it sort of depends on each individual drug, if that makes sense. Although 3 phases is the sort of the general rule. So I feel like that was both very specific and also very vague. But
no, that's really I really enjoy listening to that. Yeah, because is that a bit like the vaccine phases? Because then you because with phase 1, it's kind of a small group of people, then you gradually get bigger to a larger audience. Is that what you call the people who are getting it? But for the final question, when you're testing on animals, how can you be sure that animal nerves can be compared to human nerves?
So, I should explain. I've never personally done any experiments with animals. I've used cells, but I haven't got any further than that. So this is based on what I know from my colleagues who have done animal experiments. There are lots of reasons to believe that sort of mice, which is the animal sort of study I know most about, are good models for human pain. And the reason for that is because many of the proteins that we're interested in targeting with our pain medicines are also present in mice with very few modifications. The protein that I've been talking about this whole time is very, very ancient, extraordinarily ancient. It's one of it's present in bacteria, for example. And there are drugs that exist that people use for anesthetics that will bind and block bacterial versions of this protein. So in that sense, it's very old and not much has changed. So the mice model is pretty good, right? There's very few differences between the protein in mice and the protein in humans. Beyond that, mice seem to respond to pain in a similar way to humans. So we can't ask them on a scale of 1 to 10 how much pain they're feeling, but when humans experience pain, they do some particular things, right? Like if you bump your head, you rub it, right? And that's pretty much instinctive. Or if you put your hand on something that's hot, you remove it pretty quickly, right? It's like an instinct. You don't even think about it. And mice do the same thing. So, for example, one of the tests that scientists use to see if a painkiller is working in mice is you put a mouse on a sort of a glass-bottomed, like a sort of container, I suppose, with walls so they can't run away, and you shine a light on the underside of one of their paws, and that will cause the paw to warm up, to get a little hot, and when it gets hot, the mouse will recognize it and they'll lift their paw up, right? They take it away, just like you would if you held your hand like a little bit too close to a candle. So what you can do is you can take mice and give them that test and see how long it is between when you put them, or you start shining the light, and when they remove their paw. And then you can give them the drug, or whatever you want to test, and then you repeat the experiment. And if the mouse is, if the drug is working, and the mice is feeling less pain than they did before, then it will take them longer to work out that they're getting a sort of a trigger for pain in their paw. So it takes them longer to lift it up, and you measure the difference in how long it takes for them to lift up their paw. So that's one way, for example, that you can use, and we think that's a fair test because humans do the same thing. You can perform this test on humans, and it's exactly the same results. Humans that have taken painkillers will do similar things. So, in as much as we can tell whether mice are a good model for pain in humans, they seem to be, particularly because they respond in similar ways to pain. This isn't always the case. Like I mentioned, it's good for sort of short-term pain, like when you put your hand too near a candle. It's not necessarily so good for chronic pain, where it's difficult to tell what's causing it in humans to begin with, and so it's very difficult to replicate that in mice. So so there are some things that makes us believe that mice are a good model for pain. Some areas where we think it may not be so good, and we're going to continue to investigate it, I suppose, and try and make sure that the models that we're using are as close to representative of what will happen in humans as we can.
That's great. Thank you so much. We got through all the questions, and it's really interesting to see how you studied the peptide and how you use the crystallography.
That's it. Yeah,
that's it. There we go. And it's quite nice because as it ties into our chemistry lessons, and I'll tell my chemistry teacher that, and I'll see what she says. But this has been such an amazing opportunity. Thank you so much for letting us interview you.
It's absolutely my pleasure. Thank you for having me on the podcast.
So a massive thank you to Dr. Stephen McCarthy for letting us interview him. We had an incredible time learning about how peptides and spider venom could be used for painkillers, as well as how you can test the reliability of them, and how you can find out what certain peptides do. And thank you guys so much for listening. You can reach us on Twitter @TheSlightlySci, on Instagram, The Slightly Scientific Podcast, or on our website, slightly-scientific.co.uk. We hope you enjoyed today's show, and make sure to stay tuned for next month's podcast. See you then. Goodbye.